afternoon. Well, I want to talk about a little bit about babesiosis. It's produced by this babesiosis. It's a parasite or protozoa live inside of the erythrocyte. Is basically transmitted by the cattle tick, or called Rhipicephalus microbolus. The babesiosis affects mainly cattle in the tropical and subtropical region of the world, cause a lot of economic loss in the dairy and beef cattle. Well, there are no an effective method of vaccination or control of this disease. Basically, they are an alive vaccine. People used to treat animals with a parasite. Always have all these both methods have a lot of disadvantage as a risk of co-transmission, blood-borne pathogen, because the vaccine is produced by infecting cattle with the Babesia bovis, and then the blood is taken from this animal and used as a vaccine for the animal in endemic areas. This kind of vaccine need a cold chain method of transport to keep the Babesia bovis vaccine alive. There are dangerous parasites. There are a lot of problems with this kind of vaccine, and the parasite treatment is also have disadvantage because the cattle tick become resistant to the parasite treatment, and perhaps in some areas this treatment is not effective enough to control the tick, which is the vector of this protozoa. And then, well, there are several people who try to produce some recombinant vaccine. People isolate different antigens. These antigens, some of them are related with the infection of the erythrocyte. People have been trying to isolate, for example, peptide from this major protein in the Babesia. Some of them even there are some people try to use nanoparticles to produce vaccine against babesiosis. Then we decide to produce something different, like a new microparticles. Basically, in some research we have been doing in the lab, and then we decide to use polyglycidyl methacrylate because it's have been used in human. Different kind of hydrogels in human, and look like it's very biocompatible, or you can manage the biocompatibility of this kind of particles compared with other kind of microparticles. It's easy to do the polymerization by dispersion polymerization, and then we decide to go with this synthesis of this microparticle, and in order to functionalize the microparticles with peptide, that people say that this peptide work as a good induced protection against the infection of Babesia bovis. And then, well, this is more or less the synthesis, the glycidyl methacrylate. This is we create the polymer under this condition using this catalyzer, and then for the microparticles, they are around 1 micrometer of this. And then after we try to functionalize this microparticle using the click chemistry, where the acidic group or epoxy group were introduced in the polyglycidyl methacrylate. Well, this is the polyglycidyl methacrylate microparticles, and then this is after the acidic functionalization of the particles. With this, we use this group to put the peptide. The peptide have this a group here that interact with this acida group, and then we're going to have the peptide functionalized on the surface of the particles, like this one. No? And then this is the particles with the peptide. We put a different quantity of the particles, and then antibodies, fluorescent antibodies against the peptide. Then in order to see they were functionalized. They were functionalized. We can see here that in the photo. And then we use these particles, microparticles, to immunize mice. They were immunized subcutaneously, different group of animals with particles, only particles, and the different two peptide from the MSA2 protein. And then, well, no coming here, but yeah, well, we determine the titers against the peptide by ELISA. Then we can, these mice took the ELISA titers, okay. But we use the blood to do an in vitro study and see if the Babesia parasite, the erythrocyte, in vitro. Well, this is a normal sera from mice, and this is against sera from peptide mice with immunized with peptide 1, microparticles with peptide 1, peptide 2, and this is the only microparticles. And then what we saw a statistical difference in parasitemia of the erythrocyte, where inhibited statistically the infection of the erythrocyte by these antibodies. This is a photo of the erythrocyte and the Babesia inside the erythrocyte. This is fluorescent as before. We analyzed the CD4 and CD8. What we saw here that there were a good expression against CD4 in peptide 1, microparticle peptide 1 and peptide 2, but the microparticle per se produce an important induction of CD4 and CD8. This is more or less the same as before. This is my conclusion. No? We established a good method to synthesize glycidyl methacrylate microspheres, allow to functionalize and allow the click chemistry for functionalized with peptide. Half a microsphere induced CD4 and CD8. We went, it's not in the right order, we went to cattle with these microparticles using only one peptide, two peptide, and then the cattle were infested with Babesia, bobbies, but the microparticles, microspheres, couldn't avoid the infection of Babesia bobbi in cattle in one preliminary experiment. Well, we need to do more in this area. This is only a preliminary result. These are all people involved in this project. Monica from Argentina, Daniela Flores, Miriam from the IBM, Venus from our institute, Ala Liu is here too. Thank you.
So we've got time for some questions. Why do you think the microparticles are stimulating the T cells?
Sorry?
Why do you think the microparticles stimulate T cells?
I think that all the microparticles we use have this acida group on the surface. It's a good charge, introduced charge to these microparticles, and then the microparticle per se subcutaneously could be a source of stimulation to bring all the cells of the immune system to the skin of the mice in this case. We need to study that if that happened in cattle, but we try to see what happened in cattle, but mainly because the size and the charge of this microparticle could be the source of stimulation of the immune system.
Any other questions? Mark?
What's next with your development? Are you planning to do anything further in cattle or to...
Yeah. Well, if I receive a grant, I could do many things. Yeah, one thing could be we can change a little bit the arm in the microspheres in order to put peptide, bigger peptide, longer, with different sequence. Also, we can try to make a microsphere more biodegradable microsphere, less toxic. What we can try to put more peptide from the Babesia bovis, different one, at least a good 3 or 4 together, put it together in microsphere and try to do again the experiment in cattle and see if we can have good results.
Manuel, I didn't understand when you said that the live vaccine was only safe in cattle under 1 year. What was the reasoning behind that?
I'm not so expert in that, but I think mainly cattle, the young animal, they have people say that they have a T cell response that different to the adults. And then it's mainly a T cell response that protects the young animal against the babesia. But in ours, it's totally different. It's basically antibodies and doesn't have the same level of protection that when you are immunizing young animals, 6 months, something like this.